So I've already talked about protein folding, and we've talked about things we do to try to study how proteins fold. But it's important to realize protein folding in a computer, the way we normally do it, is kind of similar to protein folding in a test tube. And when you try to fold a protein in a cell, things can be a lot more complicated. Cells are really, really crowded. The protein isn't in infinite dilution. Furthermore, cells have to maintain their proteome, or cells have to maintain their number of functional proteins essentially all the time. So if something happens where the cell gets heated up and there's a heat shock, we need to make sure that our proteins stay folded. Furthermore, there's a lot of big proteins that just won't stay folded or have a lot of trouble getting to the folded state. And for that, nature's evolved its own special set of tools we call chaperones. And essentially, we have here our protein folding type diagram where we have an unfolded protein and it folds into its functional, correct, and useful shape. But inside the cell, you also have the propensity to misfold. Basically, when it's trying to go from here to here, it screws up and it ends up in a shape like this. Shapes like this are generally considered bad because not only do they not do their job, but they also stick together. And when they stick together, that's when we run into problems like Alzheimer's disease and Parkinson's disease and things like that. So what we have to help out is these guys, which are called chaperones. And the job of the chaperone is essentially to prevent this whole process from happening and kind of rescue things from here and bring them here, and also just block this to make sure that this happens.
We know that proteins assemble themselves because different parts of them stick to each other in a way that makes them want to adopt a certain shape. But the problem is that sometimes in the cell, you can have two different proteins that are hanging out in the same space and haven't folded together into the proper shape yet. And then the parts of them that might stick to themselves can also stick to each other. And the proteins can end up aggregating together. And this can create huge problems for the cell that have been implicated in diseases like Alzheimer's and Parkinson's and mad cow disease. And so one of the ways the cell prevents this from happening is by involving chaperone proteins, some of which mainly have a role in binding to proteins before they folded properly and preventing them from sticking together or aggregating. And still others may have some role in binding to proteins that have aggregated or else have misfolded into the wrong conformation and pulling them apart back into a shape that would allow them to fold again. Finally, there's one kind of chaperone called a chaperone-in, which is shaped like a barrel and actually engulfs and swallows a protein and helps it fold while confining it inside a cavity. We don't really know how chaperone-ins work yet, but that's one thing that we're working on that we think is very interesting.